Okay, immune system. A lot of stuff going on here. So a lot of it you should have ran across in your previous classes. Now some of it you've not seen before, but as far as like, white blood cells, what they do, where they come from, that kind of stuff, you should already have a general idea of how that works. But just some basic definitions here. You can read that yourself as far as how we define immunity. It's our bodily response. Okay, when bad things come in, whether it's microbes that cause disease, whether it's particles that are floating around in the atmosphere, or whether it's just things that are produced by our cells that are harmful to us. In any case, there's got to be some sort of house cleaning. There's got to be some way to protect our body from harmful substances, and that's what the immune system targets. Okay, so we do throw in abnormal cells up here. I'll talk about cancer and how our body handles that, those kinds of things. So we've got, like I said, invading pathogens, worn out parts and cells, destroys any abnormal or mutant cells. I'll talk about how our body can recognize that. We'll talk about allergies, something called autoimmune responses, when your body sort of recognizes itself as something foreign. You can obviously see how harmful that could be. As far as bacteria and viruses, keep in mind, think back. I don't know if you've all already completed microbe prior to this course, but if not, just understand a basic difference between bacteria and viruses. Bacteria are single-celled living organisms that are capable of reproducing, growing, and having their own lives. Viruses are considered right there on the boundary between the living and the non-living. They are not cells. One of the things about what defines something that is alive includes right now our definition of what a cell is. It has to be a cell or made of cells. Viruses challenge that because they are acellular, without cells, right? The reason that they challenge that is because they contain genetic material, which ultimately everything in life shares is genetic material. Not only do we share genetic material with everything else that is alive, but we share the same type of genetic material. We all are comprised of the same 4 nitrogenous bases, right? A, G, C, and T, and those bases line up in different lengths and in different orders, but other than that, we're all the same. Viruses pose a problem because they do have genetic sequences. They grow, they reproduce, they live inside living things. However, within themselves, they cannot survive without a host, and all they are genetic materials. And there's other things that, if you take micro, I don't know if you'll talk about it or not, but you'll learn, especially as of now, little things called like prions and viroids and things like that, which are none of, not even as complicated as viruses. They're actually just little like RNA fragments that are floating around that can be very dangerous, just like a virus can. So how do you classify that? That's just a little chemical floating around. At what point do you classify something as being alive and not being alive if you're looking down at that small level? It's interesting. Anyways, the thing about when you use the word virulent, that doesn't just mean viruses. Virulent means very potent, very, very active, very quick-moving in terms of the onset of disease and that kind of stuff. So when I say virulent forms of either bacteria or viruses, they can cause disease. And if something is capable of causing diseases, of course, we call it pathogenic. Okay, as far as leukocytes, remember leukocytes and erythrocytes are the 2 types of blood cells. Erythrocytes are the red blood cells, which we're not going to be discussing in detail. Leukocytes would be your white blood cells. We're going to talk about the different kinds, where they come from. I'm not going to get into as much as if we were talking about blood, but you should at least know the main types of the main 5 types of leukocytes that exist. We've got eosinophil, basophils, monocytes, and lymphocytes. Okay, now there's derivatives of all of those, and we'll discuss some of them. But in general, notice these fills, right? These fills right here are what's called granulocytes. If you look at inside of their inside of their cytoplasm, you'll see tiny little granules all in the cytoplasm. The first one, neutrophils, are kind of generic. They float around. They're usually the first things to arrive at a scene. Anytime there's an infection, anytime there's a damage, like a tear in the skin, anything. Neutrophils are usually the first ones that get there. Now, there's some that are already there, and I'll talk about those in a second. But as far as moving around the body, they're very mobile. They're all over the place. Your highest numbers, this should represent the highest percentage of your population of white blood cells. Phagocytes mean they do what? Yeah, they'll eat bad stuff, okay, or good stuff, depending on what they get a signal for. That's why it's dangerous. Now, your eosinophils are very specialized, okay? They look sort of like neutrophils, except when you stain them with a stain, or like right stain and that kind of stuff that you would look at a blood smear with, you would see that they stain kind of orange because of the chemicals that are inside of them. So they're easy to distinguish when you're looking at a blood smear. They comprise like less than 1% of all of your blood cells. So that you shouldn't see, if you take a blood sample from someone and you do a blood smear, we would have done this had we studied the blood lab, but you do a blood cell count. Okay, maybe some of you have done this in your other anatomy class, I don't know, but you go through and see how many white blood cells you see, what types per 100, whatever. If you see more than like one out of all the white blood cells present, for example, if you see more than one out of 100 that are eosinophils, that's rare. They're hard to see, they're hard to find, they shouldn't be there. Because if they are elevated, it's indicative of something. Your eosinophils are going to be elevated usually in two big situations. Number one, an allergic response. If someone is having an allergic reaction or their allergies are kicked in, there will be an elevated level of eosinophils. Number two, a parasitic infection like a worm infection, especially. Some sort of, actually they call those infestations if you have a worm as opposed to an infection. It just sounds grosser. Okay, so eosinophils have chemicals that they secrete that fight these parasites, but they end up getting activated in allergies, and I'll explain to you why a little bit later. Basophils are a little bit more common than eosinophils. They release histamine and heparin, so these are the guys that are white blood cells that are floating around, and their biggest job is to, heparin, of course, can help, you think of it like sort of blood thinning or something like that, but it makes the blood more mobile, is what it does, and allows it to get through tissues more easily. And then in conjunction with that, histamine, of course, dilates blood vessels and loosens them so things can get out of them more. That's why you get swelling with histamine release and that kind of stuff. I'll talk about that a little bit later as well. The whole point of this is to, basophils help create an area where there's more blood flow and more white blood cells can come flowing in, okay, to help fight this infection. So dilate the blood vessels, make them more permeable, let the blood flow more easily. That's what basophils are going to do. Monocytes, what they like to do is, sort of like in their immature form, they're called monocytes and they're floating around the body. But what they tend to do is, I say, change into macrophages, which are tissue-bound. Now what that means is, monocytes, like neutrophils, they're both start by floating around the body. Now neutrophils stay floating around. They just kind of move all around the body. Monocytes do the same thing, except they are unlike these two. See how these both all end in phils? They're sort of all part of the same family. These are granulocytes, so how you can tell the difference is they have little granules inside of them. Monocytes are sort of like neutrophils, except they are agranulocytes. They have no granules in them. They're floating around, and what they do is when they get somewhere, they tend to stop somewhere. Now, it's hard to say why or where, but whenever they stop somewhere, they sort of hang out, and they stay there. And when they do, they metamorph into what's called macrophages, and then they become tissue-bound. They become part of that tissue now. Okay, so it's a little bit different than it is with neutrophils. Then, of course, your lymphocytes are your B cells, your T cells. They're highly specific. These guys are sort of like the grunt work. They're kind of generic. They're not so specific, especially even your monocytes and your neutrophils, very, very generic acting. Lymphocytes, highly selective. They are highly particular in terms of what it is they like to bond with and what they target and destroy, and I'll show you why. You've got B cells and T cells. The biggest difference between your B cells and T cells is how they work. Your B cells are little factories, and what these little factories do are crank out antibodies. And what these antibodies can do is hook on to bad things and mark them and flag them so that your other cells, like your T cells, can recognize them as bad and start to act against them. So these guys are sort of like your special forces of your immunity army. Okay? They don't go in first. You let the grunts go in first to do all the work, and then you bring in the bad boys to clean up the mess. Okay? That's what these guys are doing. Now, their T lymphocytes are sort of like killers. There's a couple branches of them, but the most common type that you're familiar with are the ones that can go in there and actually grab onto stuff that's been marked and just destroy them. They can either release stuff that'll burst the cell. They can phagocytize. Well, they don't really do much phagocytosis, but they can just destroy those other cells that have been marked. Okay, so we have what's called antibody-mediated immunity and cell-mediated. The difference is, when I say cell-mediated immunity, I mean this T cell comes in contact with another cell, they lock together, and then it releases stuff to destroy that cell. Antibody-mediated immunity means the B cell never actually interacts with that which is bad. It sort of just floats around, and it produces antibodies that then react. So it's not directly linked up cell to cell. That's why it's called antibody-mediated, not cell-mediated. Okay, they come from the bone. All these leukocytes that you see here arise from the stem cells in the bone marrow, just like all of your blood cells. Okay, you will see that the reason that B cells and T cells get these letters with their names is because of where they mature. Okay? B cells actually mature in the bone marrow, so B for bone marrow, and when they are released, and then they sort of travel around and tend to collect in your lymph nodes. Okay, hence the word lymphocytes. Okay, they are produced and mature in the bone marrow, your B cells. They travel around. They tend to land in your lymph nodes, and they sort of call that their headquarters. So if you ever extract some lymph nodes, you'll see there's a high count of lymphocytes in there. They're growing, they're reproducing, they're copying themselves. And not only that, they are taking the lymph. Remember what lymph is. Okay, lymph is sort of like leftover fluid from blood and some other things, so things can pass through your lymph. The same kind of organisms and microbes that would travel through your blood also can travel through your lymph. So when that lymph is collected and passed through your lymph nodes, these guys are waiting on them. They're standing there, and that's sort of how it filters out all the bad stuff. Okay, now your T cells, I'll talk about this later, but your T cells come from the fact that they mature in the thymus. Okay, so that's where the T comes from. Your thymus, of course, is that little butterfly-shaped organ right up here. Actually, down here, right on top of your heart. Okay, you can see it right there in that picture. It's actually much larger in a child, right? And it actually atrophies as we get older. It gets smaller and smaller because it's under the assumption that we have sort of already built up our stock supply in our families of related T cells and this stuff. So when you're a child, you're forming all of that at a much faster rate, so it tends to atrophy as we get a little bit older. Okay, you'll find lymphoid tissue, of course, in a lot of places. If you study about the lymphatic system, I know you had to have talked about that at least in 102, I think. So you've got lymphatic tissues in the bone marrow, lymph nodes, spleen, tonsils, adenoids, appendix, pairs, patches. These are just little places all over your body where there's a specialized tissue that we call lymphatic tissue, where you can find these guys hanging out. Okay, I talked about this already. So where they come from, T and B, what it means. Okay, now we have what's called innate immune responses and adaptive immune response. Basically, it has to do with generic versus specialized. Okay, it says innate. Next to it, it says nonspecific. So this is not highly specific. It's sort of just general house cleaning kind of stuff. It's going to work immediately anytime there's a threatening agent that your body's been exposed to. And it's non-selective. It sort of takes what it can. It hits, it misses. It could get some good with the bad, that kind of stuff. Okay? It's a first line of defense. It's limited in its scope. Not limited in scope. It's limited in its precision, but it does wipe out a lot at the very beginning. Here's your most important two cells that are going to be associated with that: neutrophils and macrophages. Okay, they're generic. They're floating around everywhere. They're in higher numbers than everything else. They're going to come in contact with this stuff first. Okay. Now it also says along with several plasma proteins. You have a lot of proteins that are just made by your liver that get secreted out into your bloodstream that hang out and become a natural component of your blood. Okay? They're just little protein chemicals that are floating around in your blood. Now they're going to be very important. They're going to have two big roles. Okay? Number one, they can interact with these foreign substances. They can lock in with them and they can help mark them. It's called opsonization. They can come in. If these bad things are floating around, you've got just generic proteins floating around your blood. One of the things that these proteins can do is interact with that and help neutralize any bad things, sort of plug them up and keep them from binding with anything. Another thing that these proteins will do is think about osmosis. Okay, we know that depending on water will move by diffusion And you can get it to move according to where your solutes are, like inside and outside of a cell. Remember, if the inside of a cell is 99% water, 1%, let's say, sugar, and the outside of the cell is 90% water, 10% sugar, which way's the water going to flow? Think about it. If you've got more water on the inside, less water on the outside, we've got a barrier that's selectively permeable. It won't let things even out, like in terms of sugar and water. The sugar can't move. Only the water can move in that case. So what that means is the water is going to leave. Well, anytime you have anything dissolved in water and you don't have pure water, if you've got membranes there, like blood vessels, cell membranes, that kind of stuff, if you've got some membranes that are present and you've got dissolved things in that fluid, you're going to have osmosis. Osmosis will be affected depending on how many things are present, right? The higher the concentration of the proteins. For example, if proteins start getting out of the blood vessel and out into the tissue, it increases the concentration of proteins outside of that blood vessel. That means it's going to draw water out with it. So the more water that you have pumping through your blood, it's going to draw more fluid, draw more blood out from the vessels, out into the tissue because of osmosis. Okay? That's the two big things that proteins do. Affects osmosis and can help neutralize bad things floating around. Okay, you have what's called adaptive or acquired immunity. This is stuff that it takes practice for your body to get. The stuff that's involved with like booster shots and all this, we'll talk about that, but this is specific. You have to come in contact with something. Your body remembers what it looks like, and you form things against it. It's very specific in terms of what it targets. Okay, you have to be exposed to it. Now, it takes more time. This is nowhere near as fast, but it's also much more specific, so it's a trade-off. Now, these phagocytic cells that are involved with all of this, I want to point out that it's very complicated in terms of if you have a cell that wants to eat something else, realize that this cell that's eating, this phagocyte, is not a living organism in itself. Okay, it's just part of a living organism. There's no little tiny brain inside these little cells that go up to something and say, hey, this is bad, or hey, this is good, I'm going to eat it or I'm not. There's no reasoning process. So there has to be a way, if you've got these phagocytes floating around in your body, there has to be a way for it to recognize good and bad, otherwise it would eat everything. Okay? So that's why I mentioned this. These phagocytes are covered with what's called toll-like receptors. What do I mean by that? Like think of Nestle Toll House cookies, right? It's got the little chocolate chips sticking out the surface. Okay, so that's what we're talking about. Toll-like receptors are little receptors that stick out of the surface of the cell membranes. Okay, so what they do is they have the ability to move around, and when they're moving around, they bump into stuff. And if there's ever a chemical reaction whereby they can lock onto something, then that's what they're going to phagocytize. Okay, now hopefully they've been weeded out, and genetics have made all these proteins so that they do not interact. The proteins that are up there do not interact with our own cells. That's the goal. Sometimes that happens in diseases and things like that, but it shouldn't. So it's geared to not recognize ourselves, but recognize foreign proteins that are not part of the human genome, that kind of stuff. All right. Okay. Now, another thing about innate immunity, something included in that. Remember, innate is just sort of generic, very fast responsive, not specific. Everyone has it within you already. Innate means you're sort of born with it within you. Okay? It doesn't take any special exposures. Innate immunity includes inflammation. That's what I'm going to talk about here, but realize also, in addition to inflammation, interferon. If you had micro, you might have discussed interferon. I'll mention it again. Natural killer cells and the complement system. The complement system is going to be involving those proteins that I talked about that are floating around in your blood. These are all different ways, I'm going to discuss all of them, whereby we use innate immunity to help tackle these things that come in our body. We know what inflammation is. We've all experienced it at some point. But it does serve a purpose. And it's funny because you've got to achieve a balance, right? You don't want so much inflammation that it's compressing nerves, impeding circulation, causing too much pain, that kind of stuff. So instantly, you always try to reduce that swelling, but you got to realize that it's a natural, healthy response. There is some degree of swelling and inflammation that must occur to expedite this healing process. So it's about achieving a balance of what's too much. You think about cycling, right? You think about these things about you ice it and then you use a heat pack, a heat compress or whatever it is you use. So you do alternating cycles of hot and cold. And the reason is just that, to maintain a balance there. You don't want to ice it for too long and prevent all of this swelling, because that's your body's natural defense against it. But you want to ice it for a while so it doesn't swell up too much, and then you can increase the blood flow and increase the movement of that material by putting on some warmth and increasing blood flow and that kind of stuff. So that's the whole cycling part of it. But it says plasma proteins contribute. I'm going to explain that a little bit more in detail a little bit later about setting up these osmotic gradients. But what is the point of inflammation? Isolate, destroy, and inactivate the invaders. The point is to, at a localized site, inflammation means what is happening. I mean, there's a bunch of different steps. We'll talk about it, but it's not that big of a deal. You've got blood coming in. You've got more blood leaving the blood vessels. You've got more tissue fluid accumulating. Ultimately, what that means is you've got more white blood cells being recruited and brought it into that area. You've also got more blood flow so as to not only deliver the goods, but take away all the bad that's being produced. Okay? So remove debris. Prepare for subsequent healing and repair. Bring in clotting factors if necessary. Increase that. Bring in more oxygen. All this kind of stuff that our body's going to need for that area. So what ends up happening, like whether you have tissue damage or bacterial invasion at a very specific site. Now think about it. Right? Even if you scratch your finger and it becomes infected, we all use that word loosely, but you scratch it and it starts to swell, starts to hurt a little bit. It's kind of red. What is happening at the cellular level in this case? Well, the first thing that's going to happen is just an innate immune response. You're going to have inflammation. You're going to have mobilization of neutrophils and monocytes and that kind of stuff. The first thing that's going to happen is a release of histamine by mast cells. Now, what are mast cells? Mast cells are they're just very local. They stay there. Mast cells are cells that have sort of moved around and become part of that tissue. They stay there. And really, probably their biggest role and the most important thing that they have are just to hang out in tissue, and whenever that tissue is damaged, do this. They release histamine. Now, why would they want to release histamine? We know that histamine causes swelling. That's why if your eyes swell up from whatever, you take Benadryl, right? Because it's an antihistamine. Obviously, if it's blocking histamine, that's reducing swelling. So histamine must cause this swelling. What happens is the mast cells that are actually integrated as part of that tissue, when that tissue is damaged and torn and physically damaged, those mast cells are damaged. They release their contents, and some of those contents are histamine. Now, when histamine is released, what happens is right around that area of the histamine release, you've got increased capillary permeability because it interacts with the cells that make the blood vessels, causes the blood vessels to sort of, the muscles anyway, and causes them to relax a little bit. It dilates them. It allows the space between the cells that are making the vessels to open a little bit for more permeability, and stuff starts sort of leaking out of those capillaries a little bit more. Some local arteriolar vasodilation. So not only do the capillaries become more permeable, because I didn't talk about this because I didn't get to lecture on it, but capillaries don't have any muscles around them to constrict them or relax them. They just got little ring muscles every once in a while that can pinch shut or open. That's all they have. Now, the arterioles that feed into the capillaries, they do have smooth muscle, and they can constrict and they can relax and help control blood flow. So the increase, what will happen is as it moves up to the arteriolar level, it increases total blood delivery to the tissue. That's why it gets warm. That's why it gets so red. There's an increased amount of blood flow. Now, at the more specific site, we've got, because of this permeability of capillaries, you've got swelling because that which is coming in moves out into the tissue, and obviously can have pain because of that pressure builds and then pushes against nerve receptors in that area. Anyway, the point of all of this is you've got more plasma proteins coming in because more blood's coming in, more clotting factors coming in if there was a tear in the skin. There's more phagocytes get recruited and brought into the area, all of which is going to help repair that. Okay? And then what can happen is things called pyrogens. Pyro means what? Fire. Okay? Pyro in this case is going to be in reference to fever in the body. Whenever these phagocytes get recruited and they come in and they start tearing stuff down, one of the things that they release as they're doing that are little chemicals called pyrogens that get into our blood, travel through up to our brain, and get interpreted as, okay, in the hypothalamus, it's going to reset the temperature. It's going to say, okay, as a result of this chemical being released, I'm going to take the 98.6, move it up to 100.5. And then your body starts shivering to generate more heat. You're coming down with the fever now. You start shivering, you feel cold, and your body's trying to adapt to this new internally reset temperature, okay? Based upon just these phagocytes sometimes. Okay. All right. So your resident macrophages are the things that are already there. I told you, like monocytes that have traveled there and stayed there and parked it. Now they're called macrophages. Things that are sort of already localized in that area are obviously the first ones going to be able to respond. So within the first hour, those guys are going to start working. Okay? Arterioles dilate, histamine release, already said that. Plasma proteins now leave the blood and they enter the area. Now remember what I said a minute ago. When plasma proteins leave the blood, it changes osmosis. It increases the solute concentration outside of the blood vessel, thereby reducing total percentage of water in that area. So that means water is going to flow out with those plasma proteins because of its diffusion gradient now. So more water leaves. As a result, more edema, more tissue fluid accumulates. Okay, that swelling. Okay, fibrin can come in. Fibrin is going to be involved with blood clots if necessary, but fibrin can help wall off the area from surrounding tissues, create little pockets to separate it, to keep it from spreading, that kind of stuff. Neutrophils, now these guys right here, not the ones that, this is not stuff that was already there. Now, after this is all the stuff that was already there that's reacting. Now, what comes in next are neutrophils, just because there's so many of them in the body and they're constantly in circulation. They start traveling around. They get there first, in terms of the ones that are mobile. And then after them, some more monocytes end up coming in. About 8 to 12 hours later, they will swell into macrophages and actually become part of that tissue now. Okay, they become residents of that area. So it's anywhere from the first hour to about half a day. This is what's happening. All right, they adhere as these guys come in. This is neat. It's called diapedesis, right? They adhere and are able to stick. Now realize this is kind of a high velocity situation. These guys are moving through, and obviously by design, the inside of your blood vessels are pretty smooth to keep things from snagging, that kind of stuff. There's actually special molecules called CAM, cell adhesion molecules, that are specialized proteins that are sticking out of the membranes of these cells that allow them sort of like to hook onto stuff and to grab onto the wall. And when they do, they can latch on and they can start squeezing through. Okay? And so these leukocytes that are coming in are able to do this diapedesis. Diapedesis. Yes. That's squeezing through this blood vessel right here. That's what it is. And your white blood cells do that. You ever watch Osmosis Jones? He does this, right? I'll refer to that often. That movie. You got to watch it. It's a good show on many levels. It's good on many levels. But okay. Okay, now within a few hours, it says of this response, we get a lot of leukocyte proliferation. There's some chemical messages that are sent. As the phagocytes are breaking stuff down, they're releasing things that are sort of like flares, and they're traveling through the body, and they're saying, hey, something's going on here, and it basically calls for help, and it calls in recruits, and more stuff starts picking up on this. The body starts shipping more white blood cells to that area. It increases. More of them are produced and in circulation, and they end up going to this area. Your neutrophils, for example, those numbers may increase 4 or 5 times as far as your white blood cell count goes if there's an infection. Monocytes obviously are going to increase much slower. They're not nearly as mobile as the neutrophils, and not as fast. But they tend to form macrophages, like I said, that sort of stay wherever it is they land. So that's the trade-off. Now, bacteria, all the while, all these things are increasing. All that stuff we just talked about is happening. Bacteria start to get marked. So if there's any bacteria that make it through this area, and they start traveling around, they start to get hammered. They start to get marked for destruction. Now, they might not automatically at first, all of them might not get killed by anything. They might make it through, and they might start traveling around. at least into the local tissue. Hopefully we don't get too much of it in the blood. Realize most bacteria don't multiply in the blood very well. There are some that can. Most of them sort of, it's not a place where they can latch on to nutrients. It's not a place, it's kind of a high velocity situation. They want to get deposited somewhere, and then they can sort of take root and they can start to establish a colony. But anyways, when I use the word opsonin, an opsonin is anything that can stick to the surface of a foreign object, whether it's a bacterial cell, a piece of a chemical, whatever. An opsonin can stick to it and basically flag it, can help minimize its impact, help neutralize it, help mark it for something else to come and destroy. That's what an opsonin does. Okay, so it's a generic term. There's lots of things that can be opsonins. So if something is opsonized, it's been flagged. It's got something stuck to it that our immune system now can work with. This is one way, for example, I talked about phagocytes and how they have to be able to recognize self from non-self. One of the ways they do that is with the use of this opsonization. If they see something that's what's called opsonized, they can react with it as opposed to not reacting with it to make sure it doesn't destroy our own cells. Okay. Let's see. You already know that. Phagocytosis can destroy things. Now, there are a lot of chemicals that are released. This by no means represents all of them, but here's some of the most common ones. When you have phagocytosis happening and cell eating is occurring, there's a lot of chemicals that tend to get released, depending on the type of cell, the location, that kind of stuff. Nitric oxide. This can be released from macrophages, the monocytes that have been converted. It's very toxic to microbes. That's why it would be released. However, also, nitric oxide is a vasodilator. Okay? It's actually the key component to Viagra. So it does sort of a double whammy here. It's going to be toxic to microbes and also allow more blood flow to that region. So we get a double benefit. Lactoferrin from neutrophils that are traveling around. Now, hear me when I say this, because I don't know how hard it'll be to find the answer to this question. Book. Lactoferrin prevents iron uptake in microbes. Now, why is iron important? Iron is needed by bacteria to effectively reproduce. They do not divide very well and very quickly unless they have enough iron present. So, related to this, not only is lactoferrin produced that prevents microbes from uptaking iron, our body also does something else with iron. Our liver sequesters it. In other words, it hides it. It reduces our iron count. It lowers the amount of available iron in our bloodstream. And as a result, there's less for the microbes to come into contact with. Okay? Now, make sure you make note of that. All right. Histamine, we already talked about that. Kinins are sort of a generic group of chemicals. There's a lot of things they do. They activate the complement system. I'm going to talk about the complement system in a minute. They reinforce the actions of histamine. They help phagocytes migrate. They're sort of like, you remember the word chemotaxis? What that means? Taxis means to move, chemical. It's some sort of chemical that's released that causes movement towards it or away from it, depending on how it works. In this case, it's going to help phagocytes migrate to this area. It's going to be a message, a chemical that belongs to that group, and activate pain receptors. That's interesting. We really don't understand how this pain pathway works in the body. And even the drugs that we use to suppress pain, like Tylenol and all that kind of stuff, we don't. I'm going to say this loosely, but we don't really know how all that works. We just know that it does work. They prevent the formation of what's called bradykinins, which belong to this group, and prostaglandins. I'll discuss all that a little bit later. So we know that they affect those two types of chemicals and as such help minimize pain, but we don't really know how those chemicals are involved with pain. That's what I'm saying. Anyways, we have within us endogenous pyrogens. I already mentioned this. Pyrogen means start a fire, right? So in other words, a fever. Endogenous means they're within you. They come from cells that are already part of you. So the phagocytes that are in your body, as they start breaking stuff down, release these pyrogens and help get your fever going. which is another, controversial topic. Should you allow a fever, at least for a little bit? Should you always, because I know plenty of moms especially, who just want it, if their kid's like 1 degree over, they flip out and they think they should, try to lower the fever. Or is a little bit of a fever healthy, and to what point, and how do you judge that? Because you got to realize that a fever is a natural bodily response that really is the best treatment that you can have. I mean, whenever your body's dealing with it itself. Maybe we can talk about that a little bit later. Anybody work in a pediatric office or anything? Yeah, but I was going to say there's always a protocol. I just wondered because, it's funny though, because whenever you call the pediatrician, there's the people answering the phone. It's like there's a whole list of questions, and it doesn't matter what you say. They just have to go through their questions. So they ask this, and whatever you answer, they go this, and it's like, just stop and think for a minute, please. I got a question for you. Different. All right. So there's a couple others. Just be able to recognize some of them and that kind of thing. I'm not going to make you write an essay question about any of those. All right. So here's an example of like opsonization. Here's, say, a bacteria. It's been opsonized. In other words, here's a complement protein. All that means is I'll talk about that in a second, but it's one of those proteins that are floating around in the body, produced by the liver. It got stuck to a bacteria, and that bacteria is now what's called opsonized. It can float around, and it can interact with a molecule now on the surface of a phagocyte. This phagocyte was just sort of floating around. If it would have come into contact with this, it wouldn't have done anything because it's not going to eat anything. It's just a molecule there. But if it ever comes in contact with that while it's attached to something else, it'll know that it can eat it. Okay, so it's very specific. That receptor is very specific to that particular opsin, that particular protein right there. Okay, another thing inflammation does, it can help repair tissues by bringing in all the necessary factors to do so. Some of the repair can be perfect, depending on what all was damaged, obviously. Scar tissue, where you're going to get scar tissue is the replacement of cells that are non-regenerative. Your muscle cells, your nerve cells, that kind of stuff. All right. I mentioned a chemical here called salicylates and glucocorticoids. Now, these are fancy terms. We'll talk about more of these when we get into, especially the endocrine system. But I mention these because they are used to help suppress the inflammatory response. Salicylates are a chemical group to which, like, aspirin belongs. Okay? Glucocorticoids, these are what's called your cortisol steroids, cortisone, cortisol, all those names that you hear like that go with this that would be used to treat inflammation. But realize what they do, and I'll come back to this in a second, and you'll see what I mean. But realize what these drugs do is they actually suppress the inflammatory response. Now, in order to suppress the inflammatory response, in general, what you end up doing is suppressing the immune system. So there is such thing as too much of a good thing, right? Everything's in moderation. People get medicine happy, and they start taking medicine for long and taking medicine to treat the side effects of their medicine, and then more medicine to treat the side effects of those medicines from the other medicine, and then there's no point. Because, like, for example, too much of this, especially when you think of your cortisol steroids, and again, I'll explain it in a second, but you're taking it to help inflammation, but at the same time, what's the trade-off? Because you suppress the immune system, you leave yourself more vulnerable to infection, you're more tired, there's a lot of other things that go along with it. So, anyways, salicylates help decrease histamine release, think of like aspirin and that kind of stuff. And the biggest thing is, like, if you think about why you would take like Tylenol, that kind of stuff for fever. They also reduce fever by inhibiting the production of prostaglandins. Now, you're going to see this word again. And prostaglandins they're a large group of chemicals that we really don't know a whole lot about how they work and the mechanisms by which they work. But they're involved with a lot of important things, and people are trying to figure out exactly how all that plays out. They're certainly involved somehow in the pain response. Don't know how. Somehow, mostly they're like a communicator. They're involved in the communication process somehow. And by inhibiting them And by decreasing their production, okay, you can reduce pain, because if there's no prostaglandins there, then there's not as much pain messages being sent. In this case, the same for inflammation. If you can reduce the number of prostaglandins present, somehow that communication doesn't lead to as much inflammation. But again, the actual pathway by which that occurs is still a little vague. Glucocorticoids suppress most aspects of the inflammatory response, but also destroy lymphocytes inside your lymphatic tissue and reduce your total antibody production. So, okay, I want to mention interferon. Interferon is another. We've talked about inflammation. Now the next thing is interferon. Interferon is part of your innate immune response. It too is rather generic. However, we're talking specifically about viruses here. Is where you're going to come into interferon. Basically, the way it works is, without just spitting that to you, if you have a cell, your DNA already codes for all the possible genes that exist, right, inside each and every cell. Your DNA in every cell is exactly the same. It's a matter of which genes are turned on or which genes are turned off in that cell. It is theoretically possible to take an eyeball cell and change in terms of flipping the switches of what genes are on and off and let it grow into a liver. Okay, now, the biological feasibility of that, of course, we're not there yet. But certainly, the genetic capability is there, because think about it. You started out as one cell with one copy of DNA, and then every cell after that had to have had that exact same copy. At some point, there was specialization. At some point, there was control over what genes that are present are flipped on and what ones are flipped off, and that helps cells specialize and become different types of cells. So anyways, within all of your cells is this gene that is turned off that codes for little, what's called antiviral proteins, little tiny proteins that can help destroy viruses. And that's interesting. You know, where did that come from? Where did that gene come from? How did it? Anyways, so what you have is a cell, and if a cell gets infected by a virus, you got to understand this is a war. Okay? Any immune response that you have is a war that is declared between your body and that army of things, because it's never just one thing that comes in. Right? If it was just one virus and one bacteria, it would be easy for our body to handle. It doesn't work like that. Especially viruses. Those are very dangerous. When you have one little tiny virus that infects a cell, realize what it does. It hijacks the entire cell. This is why it's weird, because how is it living? Is it non-living? Because what it will do is get inside and steal all of your machinery. It's sort of like hijacking a plane, right? You use it for your own purpose. Well, this virus will come in, it'll land on your cell, it'll inject its genetic material or get absorbed into the inside of your cell, and then its genetic material will start floating around. And when that genetic material floats around, guess what? It can go to ribosomes. The ribosomes, it can go to the ER, it can go through all the same stuff that we have in our cells, and instead of cranking out proteins that our body uses, it cranks out more little viral proteins. And before long, it all gets assembled, and you started with one end, you got hundreds, thousands, who knows how many that build up inside this cell. Kill that cell, escape from that cell, and now you started with one, and now you've got like a 1,000-fold increase, and they can go out and do the same thing. So you can see how these things spread and how they're so dangerous. Another thing that makes viruses so dangerous and so hard to kill a lot of times, like the flu virus and the AIDS virus, well, just with AIDS and the flu both, there's many, many reasons why they're hard to cure. But one of them would be when this virus gets inside, remember we sort of have a squishy membrane around our cells. When this virus gets inside and it hijacks our cell and it pushes against the outside and sort of buds out. It doesn't explode our cell necessarily. It's sort of a bunch of them start pushing out. And when they do, it's sort of like if you can imagine escaping and pulling a cloak around you. They push out our membrane and they pull the membrane around them and it pinches off behind them, and guess what? They're floating around in our body with our plasma membrane wrapped around them. So how are you going to target that without targeting your own cells? You can't just make a vaccine that targets that virus and effectively get rid of it without interfering with our own cells and destroying our own cells. That's one reason. So what you'll find is, and plus viruses are so small, the chances of anything bumping into them are drastically reduced as well. So what you'll find is a lot of the treatment for viruses and things like that, they have to be dealt with in the cell. Whenever they're in our cell hijacking our equipment, we've got to deal with them then. We've got them localized. We've got them confined to a small area. However, you're playing with fire because that's a very dangerous point, because now they're ready to flourish. So it's a matter of, it's a race, what it is, against, it's a war between your immune system and that virus. And obviously, modern medicine has helped significantly in terms of our favor. But what will happen is if a virus gets inside of a cell, interferon is a chemical that is gets turned on. The gene for production of the protein of interferon gets turned on. And that cell, our body cell, starts to manufacture interferon. Now, interferon does that. It interferes. It doesn't interfere with that cell. That cell is gone. It's a goner. It's going to take one for the team, though. Because when it dies, it's going to have, hopefully, before it dies, released enough interferon to go to the cells around it and give it a heads up. Because as that interferon leaves, it goes out to neighboring cells, hopefully that have not been infected yet, and then it's a race. If it hasn't been infected yet, that interferon will bind to the surface. It will instigate a second messenger system. That system will eventually make its way into the nucleus and turn on that gene that codes for antiviral proteins. And those proteins will start getting manufactured. Now, that's not interferon. That's a separate set of proteins that are now start to accumulate, and they stay in that cell, and that cell is now ready for a viral infection. And if that virus lands in it, it'll meet it at the door, and it'll start shutting it down and destroying its genetic, its proteins and any genome that's there that it recognizes as foreign, that kind of stuff. So you can see it's a stressful situation there. And that's the way interferon works. It's basically okay, I'm going down, but here, I'll give you a heads up. Here's what's going on. Now, what's really cool about this is interferon works very, very well, but it's got to get there before the viruses get there. Okay, and it's also shown to, and this is what's neat, because cancerous cells behave very, very similarly to virally infected cells. There's actually a hypothesis that cancer arose as a viral infection and got sort of changed, and the DNA got incorporated and that kind of stuff, and now we've got cancer as we know it. And there are some viruses that can cause cancer, right? This whole HPV thing. We know that 70 to 90% of a lot of these cases can be attributed to like 4 strains of the HPV virus. So certainly there's a relationship between viruses and cancer, but there's no absolute proof that one leads to the other all the time. But interferon also works on cancerous cells. If cells are, especially virally induced cancerous cells, if they're starting to act abnormally and that kind of thing, you'll see the interferon gets produced and acts out and helps prevent sort of cancerous spread. I mention this because when interferon was first discovered, they thought it was a cure-all. They thought it was a wonder drug. And they would inject it in people and say, hey, if this works, why doesn't it just go around our whole body and do all this for us? Well, the problem is interferon is a local chemical messenger. It is not designed to make it all the way through the body. It only works, it spreads out from one little place and just goes to a few cells. It breaks down and it gets destroyed when it's injected in the body and has to go a long way. So it didn't work. In theory, it would have been wonderful, but it's not been working out. Anyways, that's how interferon works. Okay, we've also got natural killer cells, which are a generic type of immune cell that are kind of floating around. You can classify them sort of as a white blood cell. They're different than the ones we've talked about. They pretty much have one role. And these natural killer cells, they destroy virus-infected cells and cancer cells on first exposure to them. This is not acquired. This is innate. It recognizes abnormalities. It's generic. It doesn't catch them all. But anything that's abnormal because of virus productions, anything that's floating on the surface of this cell, parts of viruses that are made it to the plasma membrane, abnormal proteins because of cancer mutations and that kind of stuff, if it recognizes it at foreign, these natural killer cells will grab these cells, cancerous and viral cells, and destroy them. Okay. That's a summary of interferon. You can go back and read that yourself. All right. I mentioned the complement system. Complement is just that. It complements everything that's going on, but it's made from these proteins that I said were floating around, some of the proteins that are floating around in the bloodstream. So proteins are produced by, plasma proteins that are produced by the liver. They get excreted by the liver. They're floating around through the body. It's very nonspecific. What will happen is It can get activated if there are certain little chains of stuff sticking out on the surface of microbes on their plasma membrane. And if these guys bump into those, they can recognize them and activate them. They can activate these proteins that normally were inactivated at that point. And when they do, they can activate what's called a complement system. Some of these proteins can start working together to reach an end that is destructive to the foreign thing. These are C1. I see. I'll list C1 here, C2. There's several. It goes all the way down, a whole bunch of Cs. Unfortunately, it is not named C1, C2 in the order of which they occur. They're actually named in the order in which they were found and identified, so that makes it confusing. But in any case, C1 could activate C2 and so on and so forth. It's a cascade. In other words, one's sort of like a domino effect. One gets activated, which in turn activates another, which activates another. There has to be an initial event, though. That way we can control it. Everything just doesn't get activated at once. Now, when this happens, when you get to C5, the complement protein, what's called number 5, just it's been identified by a particular structure. C5 through C9 can assemble into what's called a membrane attack complex. It'll go on to like a bacteria, for example, and insert itself. Here's some protein, like some channels, for example, maybe on the outside of the membrane of that bacterial cell. What it will do is it'll come together and it'll wedge itself down and chemically bond to the things that form that channel, and that way they wedge it and hold it open is what they do. It's sort of like well, that's just it. It's a wedge. It'll wedge it open and everything can come in. It ends up swelling up. Due to osmosis, stuff starts flying in here. This cell will eventually kind of swell up and could explode. It helps kill things like that. It's very nonspecific, but it's there and it's floating around. What I hope you can see is that this is, in our body, all of this stuff happens at the same time. There is not one thing that happens and then it's done and then something else happens when that's done, something else happens. No. All of this stuff is concurrently happening all at the same time and working together. Otherwise, there's no way we could tackle the problems that our body faces every single day. Okay, these complement proteins are also going to have some other functions. They're going to be chemotaxins. Remember, they're going to be messengers that are sent and draw other white blood cells and other things towards that area. And they can be opsonins. These same guys here that can help make this up, at the same time, could just be opsonizing something. They could stick on the surface of one of these microbes or foreign particles or whatever, and as it's floating around, now it's flagged. Okay, adaptive immunity. This is a little bit different. We're shifting gears. We're moving from generic bodily response to highly specific, upon exposure, you get practice with it types of responses. Okay? We've got antibody-mediated and cell-mediated. Basically, antibody-mediated means we're dealing with B cells. Cell-mediated means we're dealing with T cells. Okay? Okay, I already said this. B cells mature in the bone marrow. The thymus is where T cells mature. Now realize both of them are produced in the bone marrow. Okay, all your blood cells are going to be produced in the bone marrow. But the T cells that are produced in the bone marrow are immature. And they travel through the bloodstream and they get lodged in the thymus. And only after they go through a rigorous training program in the thymus are they able to be released as functional, developed, mature T cells. I like the class work that they employ and the grading scale that your thymus uses. Thinking about implementing it myself. Either you pass the test or you die. One of the two. Okay? If you cannot, if you can't pass the thymus test, that means you have the ability to not discriminate. You can't discriminate between the body cells and the foreign cells. So you should be very thankful that that's the grading scale that our thymus uses. But I do like it. I'll run it by the board and see what they say. That's right. As long as it's 60% or better, you're in good shape. 59.4 is all it takes, and death becomes you. Okay. Now, there is a hormone that's released in your thymus called thymosin. Thymosin is responsible. You don't have to know how it works, but what it is responsible for is maintaining T cell lineage. In other words, there are different families of T cells that exist. Now, they're all T cells, but realize T cells have to be very specific because they only bond with certain things that they are exposed to. So what you do is sort of like if you had a family tree, and you started at the top, and you had the mother and the father, and then all the siblings, and then all their kids. This is a lineage of T cells. You start with things that are produced in the bone marrow, and then they come in contact with other things, so they sort of change a little bit. And then before long, you've got this whole hereditary map of, the T cells that are part of this family, the T cells that recognize this, the T cells that recognize this, and you've got this whole lineage that has to be maintained and controlled and pruned. I wish we could prune our family trees. But it doesn't quite work like that. Or I guess in my case, my family stump or shrub. I get one person said a family shrub, was it a shrub? Bush, family bush, because there were so many outward branches. It doesn't go up, it just keeps going out, all third, fourth, fifth, sixth wives kind of thing, and it just keeps going. Not with my family, but someone said that before. Anyways, now we introduce the term antigen. You have antigens, you have antibodies, I almost said antibiotics, antibodies and antigens. What's the difference between them? Just look at how you break the word down. Antigen. Gen means to form, to produce, to create. Antigens cause the creation of antibodies. They generate antibodies. So that's how they get their name. So antigens are anything that's going to cause our bodies to produce antibodies. Okay, so they're usually foreign substances or whatever. Sometimes it's ourselves, but that's bad. Okay, now there has to be these antigens so that our B cells and our T cells can recognize them, and that's what they're going to use to sort of pilot what it is they're going to be doing. All right. So I already summarized this. You can look at this yourself, but everything starts in the bone marrow. B cells actually mature in the bone marrow. T cells leave the bone marrow, go to the thymus to mature, and then we're done. Now remember, B cells are going to produce antibodies. They're not going to bond directly with anything. The antibodies will, but the B cells won't. T cells are going to actually help stick right to things. They're going to be cell-mediated, not antibody-mediated. I told you about my clock watchers, didn't I? Where's the quote? Attention clock watchers, time will pass, will you? So just keep that in mind. All right. B cells, like antibody-mediated immunity. This is amazing. This blows my mind. How our body does this. We take this for granted so much. We just sit around and you don't even think. If you understood everything that I say, and you studied even more and understand the specific details of how all of this works, and you ever get sick, you can't help but thinking about sitting there, what is my body doing right now? This is amazing. It's so cool. But anyways, all right. A given lymphocyte, so your B cells and T cells, we're going to focus on B cells here. A given lymphocyte has receptors on its surface to recognize only one unique antigen. Do you realize how many possible antigens exist in this world? An antigen is anything that could be foreign particle that could cause our body to recognize it as foreign. How many foreign things are there out there relative to our body? There's tons and tons and tons. But every lymphocyte, any given lymphocyte, the type of lymphocytes that you have, has receptors on its surface to recognize just one. So how in the world could we ever have enough lymphocytes to recognize everything that's possibly bad? It's really cool how this works. But all right. Before we get there, just how these B cells work in general. What antigens do is they will interact. Antigens, these foreign things, they're floating around. They will interact with your B cells. And when they come in contact with your B cells, what your B cells then do is turn into a factory. What they do, they turn into a factory that's going to produce and cause the existing B cells that you have to divide ridiculously. They're just going to start dividing like mad. And they're going to divide into two different types of cells, okay? Called plasma cells and memory cells. We'll talk about this. Let me see if it's in the... Okay, we're not there yet, but I'll show you here in a second. Now, plasma cells are... So you have B cells, and what happens, and I'll explain this in a second with a better picture. But when they divide, you've got this pool of B cells that you have, and when some of them get activated, they start dividing. And when they start dividing, like say one of them divides in two, now you've got two. That one is gone, it's split into two, now you've got this one. One of them might stay that way to be a memory. It'll stay there. It'll be what's called a memory B cell. The other one could divide even more and make a whole bunch of what's called plasma cells, which load up on endoplasmic reticulum and are protein factories. They crank out all these proteins that we call antibodies. Okay? And they're going to start pumping out these antibodies that react with whatever that antigen was, and they're going to start floating all around the bloodstream. All these antibodies are. Okay? I'll show you in a second. Now, there's a whole bunch of different types of antibodies that exist in our body. There's a class of 5 different ones. You will have to know these and sort of be able to probably, I don't know, match the type with an explanation of it. All right. For example, an easy way to remember all of them is the name MAG, okay? Because you've got M, A, D, G, and E are the different types of antibodies. Now these are just classes of antibodies, right? It's just sort of like a family that exists. There's 5 different families that exist. IgM. Let's start down here with IgG. Okay, this is because it's the broadest, it's the biggest. IgG is the most abundant antibody produced in large amounts. Ig stands for immunoglobulin. These are all immunoglobulins. In other words, proteins associated with the immune system, okay, antibodies. IgG is the most abundant. It's produced in large amounts. It's very generic. It can opsonize things. It is mostly responsible, its presence is mostly responsible for where the water goes. Like if it moves out of the blood vessel, water will follow it, that kind of stuff. IgM is actually our antibodies that don't that don't get secreted into the bloodstream, usually. They're actually part of the B cell. They stay hooked on to the B cell and sort of stick out like forks all around the B cell. Okay? So those are IgM. Their most important role is to be receptors on the B cells. Okay? IgA, grocery store. Found in the, what does that stand for? International Grocers Association? You know you live in a real metropolis when that's your grocery store. Huh? That's right, mine too. All right, IgA is going to be found in the secretions of your digestive, mucous linings, respiratory, genitourinary. These places is where you're going to find the highest abundance of IgA antibodies that are going to be associated with those types of conditions. So you can monitor those levels and that kind of thing. IgD on the surface of many B cells, but don't really know what they do. But they've been identified, so here they are. IgE, however, protects against parasitic worms and is the antibody mediator for common allergic responses. So what you're going to see is, remember what cell I said was associated with that as well? Eosinophils. In a parasitic worm infestation or an allergic response, you're going to see 2 things. You're going to see elevated levels of eosinophils, and you're also going to see a high level of IgE antibodies. Okay? They tend to be the ones that get activated in this process. Now, obviously, at first, so you should be able to distinguish whether or not it's an allergy or a worm infestation, but not just by this test. If you just did this preliminary test, you can probably efficiently narrow it down to at least 2 possibilities, one of those 2. Now, those are drastically different from one another, right? So then you would do more tests to figure out if it was like a parasite or whether it was just an allergic response. Okay. Now, what do these things sort of look like? They actually all look similar to that. They're sort of Y-shaped. These are just chains of chemicals bonded together. It's got heavy chains here and a couple light chains on the side. This is where it's going to be anchored to the host cell, like the surface of the B cell or whatever, or any type of our own cell will be anchored down here. These forks sticking out up here is where they would bond to antigens. Okay? And the chemistry here at these points are very specific antigens. Okay? So I talked about heavy and light chains in the shape of a Y. We've got tail regions down here. That's going to be the functional property. Basically, you're going to classify whether it's M, A, D, G, or E based upon the sequence of chemicals that you get down here. Okay, now, you guys will never have to run into that, obviously, but just know that that's how they're identified, based upon the chemistry in this region right here, because the rest of it could look very similar. Okay? Up here is going to be the specificity of that particular antibody. What will it interact with? Now, here's a lot of different antigens up here. Not all of them fit. Only that one does. Okay, so each one of these antibodies that you see are very specific. All right. Now, I'm just going to go through these pictures to explain that slide. Now, these antibodies, when they are released, okay, now some of them are generic. Some of them are highly specific. I mean, they're all very highly specific, but I mean, they don't have to necessarily be always locked onto the B cell and that kind of stuff. They could just be produced, and when they're produced, they are released. But remember, they're released in response to something very specific. Okay, so if it's floating around as some sort of bacterial toxin that caused the release of this antibody will be released, and one of the things it can do is neutralize it. While we're waiting, now this could be neutralization / opsonization. Okay, but what it can do is float around and lock these bad boys up so that they don't go somewhere and wreak havoc on our bodily tissue. They sort of deactivate them chemically by bonding to them. Then we can phagocytize those particles or whatever as they're floating around. You can also have agglutination, which is stuff sticking together. Right? Do you ever do blood typing? You did that lab. I don't know if anybody's ever typed their own blood before. One of the things you look for is, agglutination. Does it sort of clot up a little bit? That's what we're talking about here. Notice how that would work. If this is an antigen on the surface of this cell, and these antibodies are specific to that antigen, as they start floating around, they can lock one arm can lock on one cell, one arm can lock on another cell, and before long everything gets clumped up and you get agglutination. That obviously slows down tremendously where these things can go. It helps localize it to an area and that kind of stuff. Now, this could take a sort of a worse effect, I suppose, if it got heavy enough. I guess it didn't show it, but I'll go back a couple. Worst case scenario, if these were heavy enough and got a big enough lattice structure, they could precipitate out. And you could actually get a solid precipitation out of that. You could get, the antibody could cause the complement system to get activated. Here's an antibody that floated around. It's specific to these antigens sticking out the surface of that bacterial cell. As it's floating around, there's a special bonding site by which the a complement protein could bump up against it. If it does, it'll activate it, and before long, now the complement system is activated, but before it wasn't. A membrane attack complex could come in, the C5 through C9, wedge in a little hole. Before long, it could destroy that bacteria. So there's all kinds of things that can start happening here. We can enhance phagocytosis by having all of these. Now realize, remember what I said about these bases, these tail regions. They are made to be recognized by our body. The fork regions are not. So these fork regions, if they start going all the way around and bonding to different areas on a bacteria, now our body can recognize these tails as self, and they can help phagocytize. Now, don't get confused. We don't want to teach ourselves to recognize ourself. It doesn't recognize ourself because the chemistry of this has been changed. If these guys were just floating around and this bacteria wasn't here, and this cell receptor right here came in contact with this tail, it wouldn't do anything. Okay? If this was sticking out somewhere off our own cells, it probably wouldn't do anything. But when these forks bond with this changes the chemistry of this region right here. And as a result, it changes the chemistry of the whole antibody. And if the whole chemistry of the antibody changes enough to where we can still recognize it, but you get a lock fit right here, now we can recognize this as foreign. It's different than what it was to begin with. So what will happen is this part will stick to it, this part will come up here and stick to that, and before long the whole cell wraps around it, and it makes it easier to phagocytize. Okay? It helps it easier. It's easier to identify that. I'm just trying to finish these. Stimulation of killer cells. These are generic little guys that float around. Okay? They're just killer cells. They release stuff that explodes cells. Sort of like phagocytes. They're generic, but they just work a little differently. If that antibody is present and it's stuck to a bacterial cell like this, that exposed tail region then can now interact with a killer cell that's floating around. If it bumps into it, it'll lock onto it. It'll initiate a set of events inside this cell. It'll secrete something, causing it, like perforins or whatever, to go in there and bust it open and destroy that cell.